Hi everyone, how are you today or this week? Are you surviving of the exam week? Okay, so let's try to wrap up. So the exam will be up to this point, today's lecture point. So probably at least a couple or maybe more than exam questions might be out of today's lecture. So that might be a little motivation for you to bother to come here today's lecture. Okay. So let's first last lecture I brought up some membrane protein. One is integral and the other one is a peripheral protein. Why? If you thought about it at the end of the lecture, why that guy bothered to bring up such a confusing concept about two different proteins? Because these two proteins has a different track, the route to come to be delivered to the membrane. That's something that is somehow related with the organelle's function that we are going to take a look at. That's why. Okay. So let's take a big picture. Regardless of some those little miscellaneous details, I always emphasize that those little details are not really important. Okay. To understand this big concept, to be able to understand, is the main thing. As long as you are capable of doing that, that's you're okay. So those two proteins. One is, why am I making such a big deal of some so-called integral protein? Why some protein are stuck into the membrane? What is this? The feature that the fact that some proteins are embedded in the cell membrane is such a big deal. Who cares? It's the first line of the question that we are going to approach. Why? Why certain proteins? Who cares? I mean, yeah, so be it. Some proteins are embedded in the middle of the membrane. Some proteins are somehow like associated, only attached to the cell surface, not being able to. Those proteins, the reason why those proteins are not embedded in the middle of the layer of a cell membrane, but simply surface scratching, is because they are not able to get inserted into the cell membrane. That's the concept. Why? And the question is, why is this such a big deal? Why is it important? Because proteins in nature, naturally, the proteins, if you understood the proteins, the structure, proteins are usually not supposed to be easily inserted into the cell membrane because, as I said, there are two different kinds of, only two different kinds of molecules in the whole universe, if I'm exaggerating. One is hydrophilic, and the other one is hydrophobic. The hydrophobic molecules are water-hating, non-polar molecules, just like lipid, oils. Oils, lipids are all hydrophobic molecules because they are non-polar. They are not charged. They don't carry any electric plus or minus charge. Those non-polar molecules. And the other group is hydrophilic. The reason why they are hydrophilic means they don't have any problem in associating with water. They are easily dissolved in the water because they are charged. They are polar molecule. And proteins in nature, when the protein structure is finalized, usually, the overall final shape of a protein is made almost in most of the cases, protein's final shape is determined automatically by the law of the physics. Okay? Because the surface, proteins tends to have some hydrophobic surface whereas as well as hydrophilic surface. And then those hydrophobic surface tend to fold inside. Why? Because they don't want to get exposed to the water because they are hydrophobic. That's their natural law of physics and chemistry. And that's why only those area of a protein that you get to see on the surface in the finalized form is usually hydrophilic form. That is extremely important because if you mess them up, then sometimes you suffer from grave consequences, such like sickle cell anemia. Actually, those, if you heard of the sickle cell anemia, the disease, it's the cause of the sickle cell anemia disease is this misfolding, protein misfolding. Okay. The hydrophobic surfaces are exposed, so they panic and they do something about it. If you are a little more interested in, you go back to the chapter 3 lecture and then you find out because that has been explained there. Not for this upcoming midterm exam, but just for your own 교양. Probably you might be able to. But anyway, so these proteins are usually tend to be those areas, surface areas are all hydrophilic. That means they're not usually supposed to easily be able to get inside of the cell membrane. Nonetheless, some proteins have no problem with being inserted in the middle of a cell membrane, and that is thanks to those proteins we are able to enjoy all those different traffics, the material traffics. Otherwise, cell membrane would have been left bare without having any such transport protein or receptor proteins that we need for various cellular activities. Okay. The reason is they do have some very peculiar, unique structure that we call this alpha helix, transmembrane alpha helical structure. Okay. Only that much. Yeah. Although I have far more that I'd like to say about this so-called transmembrane alpha helix, but that will be just enough. Let's stop there. Okay. So you don't really have to remember this, whatever this transmembrane alpha helix or whatever, but simply put, oh, okay, fine. Some proteins have no problem of being embedded in the cell membrane because they have some special structure. But guess what? What kind of a special property of this particular alpha helix, whatever this transmembrane alpha helix, in terms of chemical properties? What do you think? You can guess. You should be able to guess. I mean, what kind of nature, what kind of chemical property enables this so-called transmembrane alpha helix? With that, by virtue of having this transmembrane alpha helix, these proteins are able to be inserted in the membrane, which means, like I said, as opposed to what I have just said, that proteins usually, the surface of proteins are usually hydrophilic, probably something opposite to that property is, even though this transmembrane alpha helix is fully exposed on the outside of a protein, but this transmembrane alpha helix contains plenty of hydrophobic area so that they are able to associate with hydrophobic membrane. Always likes tend together each other. I mean, hydrophilic molecules tend together each other, one another. Hydrophobic molecules also, on the other hand, tend together each other, but repel. Like if some hydrophilic molecule try to get inside, then who are you? You are not one of us. Get away from. Same thing, hydrophilic molecule. If a hydrophobic molecule try to sneak into the whole surrounding hydrophilic molecule, and this hydrophilic molecule have a big problem, energetically speaking. But this protein must be in the hydrophobic layer. So in order to solve such problem, this protein has solved the problem by having somehow this hydrophobic surface. See, I'm one of you, so let me in. So it doesn't have any problem. That's it. Okay. So let me move on from. Okay. So this so-called these type of a protein called integral membrane proteins. Unlike those surface protein that we get to call the peripheral membrane protein, in their origin, they are quite different in that all these integral membrane proteins are all of them are made from, because regardless of their origin, all the proteins are made starting from on the ribosomes, but there are two different types of ribosome in the cell. One ribosome, one group of ribosome, one kind of ribosome are attached on the surface of something called ER. What's ER? Where are Did I miss some ER picture? Okay, yeah. Like for example, let's say if this is the simulation of the surface of the nucleus, having various nuclear envelope, okay, then there are a whole bunch of ribosomes. Their functions are protein synthesis. And these are the model, computer-generated model of ER. Its full name is the endoplasmic reticulum. They are all membrane-bound little sac-like structures, okay, that we are going to take a look at a little further detail. So this is what we call ER, endoplasmic reticulum, shortened form of the ER. And these are another membrane sac-like structure called Golgi apparatus. And now those are mitochondria. But these don't look like a typical mitochondrial shape that you get to see in your textbook version if you had ever have any kind of a biology textbook. Okay, so that's what I'm saying. Usually, kind of there are always like some stereotype in the particular shape or structure of the cellular internal structure. These are more actually more realistic form of mitochondria than those you happen to saw in the past textbook. And these are all network of proteins. Just like your road in the city, it behaves the similar function. We call those cytoskeleton. It also provides the function of like your the bone structure like a skeleton inside of the cell. That's why. So those are some of the selected internal structure of the eukaryotic cell. And this ER, endoplasmic reticulum, and this Golgi apparatus in particular, those are the ones specifically involved in these. The topic that I'm going to just share with you today is protein transport to the cell membrane. Okay. So, all these integral, so-called integral membranes proteins are synthesized starting from this. Oh no, this is not. The surface of the ER, those ribosomes are attached, for some reason they are attached on the surface of this so-called ER membrane. And another common feature of all these integral membrane proteins are they are all glycoproteins. What's glycoproteins? Proteins having carbohydrate on the surface. And what's the importance of this carbohydrate on the protein? Try to connect. They are all the integral membrane proteins. And all the integral membrane proteins have this carbohydrate on the surface. Why? And what's the function of this carbohydrate? Recognition. So this carbohydrate attached on the protein itself, on the cell membrane, then they serve as some kind of a signal or identification by the other cells, like patrolling cells, like your white blood cells, and some other cells that are going to initiate some kind of interaction. In the multicellular protein, many different types of cells have some specific interaction to have a specific metabolism too. For that matter, correctly recognizing your partner is always very important. Without this specific recognition marker, how are you going to be able to recognize, identify, oh, this is my partner? Okay. So this is how you do it. Okay, so, and this is a typo. This is not integral membrane protein, but peripheral membrane protein, sorry. Okay. So this peripheral membrane protein, on the other hand, are those who simply associated with the integral membrane protein or some phospholipid. If you go back to, do we have any better? Okay, here, like, so if this was an integral membrane protein, which, like, by virtue of having this internal so-called what? Transmembrane alpha helix, that's how they can be stably inserted or embedded in the membrane. On the other hand, some other proteins like this, simply surface scratching protein, try to find the similar structure, same structure as this, they don't. So they can't. They can be sucked inside of this thing. So the only way they can possibly ever still be around the membrane is either try to have some kind of association with the exposed part of the integral membrane protein, or bravely enough to directly try to be associated with some part of the cell membrane. It also still depends on their particular chemical properties. Different protein will have different chemical property on the different area of that particular protein. That's the beauty of different protein structures and chemical structure things, which we don't really have to worry about in this class. So these peripheral membrane proteins, on the other hand, they are not made from the ribosome attached on the ER membrane, but there are some freelancer ribosome. We usually call the free ribosome. What do I mean free ribosome? They are floating around in the internal space of a cytoplasm. They're not particularly attached to any particular area, but simply floating around in the cell. So that's why I can compare these ribosomes as a freelancer or free ribosome. So the vast majority of other proteins are actually made by the action of this so-called free ribosome. It's only those specific proteins that are shipped, that are going to be transported in the cell membrane, or some even far beyond the cell membrane. Some proteins are even secreted protein. They are going to get outside of this cell membrane. A lot of enzymes in our the intestine and the stomach that are acting as a digestive enzyme are actually made elsewhere and being secreted and delivered into the final area. And like also you can think of insulin. Insulin is another protein, and insulin is made in pancreas and then secreted out to the blood. So a lot of those proteins are actually are destined for secretion, shipping out of the cell, outside of their original place of synthesis. And those proteins undergoes exactly the same fate as this membrane-bound protein, like I said, okay, integral membrane protein. That's what I'm going to talk today, like as the route of this protein transport for like this so-called secreted protein. And these are, so because these proteins are going to stay inside these peripheral membrane proteins, will never be exposed outside of the cell, so they don't have to have any this carbohydrate. So they're not usually glycosylated usually. Okay. That also verifies the important function of this so-called carbohydrate attachment. Hey, shut up. Unless, sorry, it's not your fault. Okay, so let's move on. So those in the eukaryotic cell, the organelles, like I said in the last lecture, most of these organelles are basically nothing but the internal membrane system, which means they are made of, though their boundaries are made of exactly same material as the cell membrane, the chemically speaking, the phospholipid bilayer that I say. Okay? So all these those you saw like ER and the plasmic reticulum, Golgi, and some other miscellaneous organelles, that they are all made of same phospholipid bilayer to have those. The only difference is they are smaller, much smaller in terms of their space area than entire cell, which is bound by this cell membrane. So that's why we usually call them as an endomembrane system, inside membrane system, which include like endoplasmic reticulum, Golgi, and there's another organelle, the famous organelle is an important organelle, which act like the stomach in your cell. It's called the lysosomes, and some other little vesicles, sac-like things, are some of the examples. So once again, you don't have to memorize all of this. Okay, these are some of the examples of the endomembrane system. That is not the point. You just simply understand, okay, there are some of these little organelles. They are all, what's the common feature of all these organelles? They are all made of this same membrane. That's it. And then we get to call them as an endomembrane system. Yeah. And they will have some different function. And sometimes some of them are also involved in the one identical function. Okay. That's it. It's not really difficult to figure them out and understand and distinguish them. Okay. So some description of endoplasmic reticulum that I wouldn't even bother to read this thing. Later on you can check yourself. Okay, so what's the endoplasmic reticulum, blah, 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 fine. Okay. And there are two different types of endoplasmic reticulum. One is called smooth and the other one is called rough. It's very straightforward. And the the way they distinguish is one is called smooth and the other one is rough, is they're outside the shape, outside appearance. For example, are there any? Okay. So, for example, this is endoplasmic reticulum, but this is an actual electron micrograph, EM picture. So some of the areas are labeled as a rough endoplasmic reticulum, and the other area is labeled as a smooth endoplasmic reticulum. Do you personally, on your own, do you see the difference? You don't? Okay, good for you. So you don't have any discriminatory mindset, which I appreciate. So if you look carefully, if you really look carefully and enlarge form, on the surface of a rough endoplasmic reticulum, if I may draw representing a surface of rough endoplasmic reticulum, then you get to see a really tiny little bit of numerous dots. Okay, and that's why under the electron microscope, to the observers, it appears that it's a bumpy, rough dot. That's why it's called rough endoplasmic reticulum. On the other hand, this smooth endoplasmic reticulum area, you don't see those little dots. That's why it's called smooth. And what makes difference in this roughy and smoothy is this little dots, right? Little dots, well, guess what? What do you think those little dots are? Ribosome, right? So those are ribosomes. For some reason, ribosomes are only attached on the surface of this so-called rough endoplasmic reticulum. That's it. So let's get back to, first let me finish quickly the representative function of a smooth endoplasmic reticulum first. Some description of a smooth endoplasmic reticulum. If I summarize the function, major function of smooth endoplasmic reticulum, one is lipid, dealing with all those lipid synthesis, including even the cholesterol. If you want to actually synthesize and provide on your own cholesterol inside of your cell, yes, that's usually in the smooth endoplasmic reticulum's jobs. But unfortunately, like in multicellular organism like human, the cholesterol synthesis is usually done by one particular the organ and then transported out to the whole different part of the body. But still, the area where in those organs, like a liver, okay, cholesterols are all synthesized and delivered. But in the liver, inside of the liver, each individual liver cells, this smooth endoplasmic reticulums are the one who actually get to synthesize all these cholesterols. So all these different kinds of lipid metabolisms are done in the smooth endoplasmic reticulum. And another very interesting thing is the detoxification too. So also in the liver, in our body, liver is the specialized organ for such things like detoxification, especially when you had a hangover, like after the heavy drinking, your liver gets so busy because of this action, the action that required. And detoxification, converting all those toxic chemicals into some non-toxic chemicals. And also storage, calcium storage, which is important for animal, and it's not only in the animal, because this calcium is important in several different the signaling, including this muscle contraction. If you want to do this action, like, oh, I want to grab this camera, I don't want to mess with. This kind of action is all done by your will, but it has to go through this calcium inside of your muscle tissue. The calcium has to be released to do so. And those calciums, before you actually initiate this action, your calcium in the cell, this muscle cell, is all stored in the smooth endoplasmic reticulum. And then once you start this action, the calciums are first all released from this ER. And that's how you will be able to do these actions, right? Okay. So anyway, speaking of this detoxification, so many different elements, chemicals, are converted into something so-called consider harmless. Then the general rule of this detoxification is actually, once again, if cell identify some foreign material you have never seen before, okay, this is something. How do they know that whether this chemical is going to be toxic or not? You don't. So you have to take some kind of preemptive action. And what's based upon what kind of rule are you going to do that? Usually, any new foreign material which is hydrophobic, then your first reaction is you consider them as a potential toxic element. Why? Because these hydrophobic chemicals that you get to see for the first time, this hydrophobic molecule can easily move around your bodies because they are hydrophobic. They can pass through cell membrane without any interruption. So if you allow them to go or escape freely inside your body, you are afraid. You never know what's going to take this. So that's why you take some kind of preemptive action. Any unknown hydrophobic molecule, you trap them. You try to trap them and then don't go away. Where do you think you are going? How can you do that? You convert this hydrophobic molecule into hydrophilic molecule. That's the basic rule of detoxification procedure. Okay? In your smooth ER, you try to convert this initially hydrophobic molecule, try to attach some small chemical residue to make them now hydrophilic, polar. Once they are done, this hydrophilic molecule cannot escape freely outside. So now at least you can make them stay where they are. And then you pick them up and then dump them, discharge outside of your system. That's the general rule of this detoxification and removing procedure. Yeah. Yes?
뉴스에서 봤는데 그 터널에서 피렌이 나왔다는 기사가 있거든요. 왜 터널에서 어쩐 피렌이 나오는 거예요?
It's everywhere. Benzopyrene is everywhere because this particular benzopyrene that I'm going to actually take as an example of this mishap in the smooth ER is everywhere, especially the burning procedure. Any burning procedure releasing smoke, like burnt meat, cigarette smoke, are the two major source for this benzopyrene. Okay. And that is one of the reasons why if you keep doing this, probably you're not going to have a very pleasant consequences. Like probably the chances of you getting the cancer, lung cancer especially, it will be very high. It's because of this guy, the benzopyrene. And also, if you keep eating burnt meat, like I do, I love burnt meat for some reason. It tastes good. Then you will get this, once again, the cancer risk will be higher. It's because of this guy, benzopyrene, the same thing. But actually, guess what? What happened is benzopyrene is, whether you know or not, benzopyrene is in original form, is an entirely hydrophobic molecule. Hydrophobic. Nothing. You don't have any charged area over here. Yes. And according to the general rule of the detoxification, what your cell does inside of your ER, this kind of enzyme, name you don't have to know, who cares about. this enzyme will work so hard to convert them, like put an oxygen over here and do. So by now, finally, you have this type of OH, like this is a little polar group. So now it became polar charged. Okay, polar, at least a polar molecule, which became the hydrophilic molecule. Finally, this is the processed version of benzopyrene inside of your cell once they are in. Now, actually, the dangerous molecule doing so much harm to us is not this, but this product. After the action of the detoxification, now we have created real monster. It can get into this internally in the middle of our DNA double helical structure and interfere the DNA replication procedure and create a general big chunk of a mutation. Okay. That's how and why. So although generally almost all of those actions taken by this ER to detoxify are actually good for us, and we should thanks to them. However, sometimes this type of mistake, they don't know what's going on, but actually their effort, the result of their effort actually is causing more harm than the original form is one such an example. Yes?
근데 그 산불이 날때 벤조피렌이 발생할까요?
Sure.
산불에서
Yes.
벤조피렌이 나오는 이유가 천공사할 때 폭파하는 거랑 관련이 있을까요?
그런 것도 관련 있고 없어. I'm sorry. Yes, a lot of those action, the burning activity will get to create this benzopyrene and right. So, okay, moving back to rough endoplasmic reticulum, because at least I have to finish this rough endoplasmic reticulum and the exocytosis today. And I wish I could have a little more time so that we can go over. If you have any question regarding those previous exam questions, then I will answer today. If you, so let me move quickly. So this, on the other hand, this rough endoplasmic reticulum's function is, one is this protein secretion, shipping out, secreting. Another thing is very important is correct protein folding, okay, inside, only for those proteins that are going to be shipped, of course. Those proteins that are going to stay inside are not made on the surface of ER to begin with. So this ER have nothing to, they don't have any business with those proteins that are going to stay inside of the cytoplasm or any, that here, this protein folding function means only those proteins that are going to be shipped to the membrane or outside of the cell membrane. Yes. And also, protein glycosylation, of course. Those proteins are going to get glycosylated, so that's what happens. This glycosylation happens here in the ER first. Okay, let's take a look at it. So, this one is also, sorry, why do I have so many mistakes? It's not endocytosis, but it's called exo. Moving out. So the process of moving the proteins that those proteins that are destined for secretion are called, generally collectively called exocytosis. On the other hand, importing the proteins or some other materials from outside into the cell is called, guess what? It's called endocytosis. Endo means inside. Exo means outside. Okay. So I get mixed up. Let's get flipped. Okay. So all these procedures start from this rough RER, by the way, is an abbreviated form of a rough endoplasmic reticulum. We usually casually call the rough endoplasmic reticulum as RER. And what about the smooth ER? SER, yes, is a smooth endoplasmic reticulum. So overview is, starting from here, the rough endoplasmic reticulum, these proteins are moving temporarily into this Golgi area, and where from Golgi they finally go outside, or some of them stay in the middle of the cell membrane, and some others are also shipped into this lysosome. That's all about this, the process of exocytosis. It goes out, or it goes, some of them stay in the middle of the cell membrane, and also some others go into this other organelle called the lysosome. The only three choices. You have option to go either go outside, or go in the middle of the cell membrane, or go inside of the lysosome. Do I have any choice? No, you don't. It's your fate. As a different protein, some proteins are already from the beginning destined for going to the lysosome. Some other proteins are destined for shipped out of the cell. On the other hand, at the same time, some others are meant to stay in the middle of the cell membrane. And we are talking about, in the exocytosis, we are talking about the transport of all these three different kinds of proteins. That's what we call exocytosis. Okay, so this is the very initial step of this exocytosis, which happens in the rough endoplasmic reticulum. As you examine carefully, slowly, take your time and imagine what's going on, and follow 1, 2, 3, 4 steps in this diagram figure. Hopefully, you can understand on your own, okay, what's going on. So first, it starts in the ribosome. Ribosome is the place where the protein synthesis is done. So once this protein being spat out of these ribosomes, the initial protein, which is unstructured, their structure, final shape is not finished yet. Because this ribosome is already, to begin with, these ribosomes are attached on the surface of the endoplasmic reticulum. This is the surface of rough ER. So even before complete completion, finishing of this protein synthesis, some part, the early part of the protein is being pushed inside of the internal space of the rough endoplasmic reticulum. So all these proteins, all this, once again, what does that mean? All these proteins that are made on the surface of rough endoplasmic reticulums are all pushed inside, pushed into the inside of this rough endoplasmic reticulum. That's it. So they are all ended up here inside. That's what they received. Three very important editing procedure. Oh, actually, there are only two. I'm sorry. There are only two important editing procedures. One is protein folding. Inside, the protein folding must be done correctly. Let me emphasize of this, the significance of this protein folding correctly done. What is the significance of this protein folding? Must be done correctly here. What if this protein folding happening inside of this ER is not done somehow by mistake, not done correctly? What's the consequence? So what? Hey, loosen up. Why do you have to be so tight? You don't really have to be always, anytime, 100% exact. Take it easy. Then what happens? Directly, its function is going to be affected. this correct folding of this protein directly affects the function of the particular protein. Always. Once again, let me emphasize, the shape dictates function. So here, this is really the only last chance for any proteins that are made on the surface and then being pushed into the ER. If they don't go through correct procedure of folding themselves, then they become garbage. Okay? Because they are not able to correctly function. If you actually let, as a ER, let's pretend that you are a supervisor of ER here, over here. And if you allow misfolded protein escape outside of this ER, then you will be responsible for much greater consequences, and you should sentence death because that's really a serious problem. So they don't do that. Once they detect any misfolded protein inside, then they all grind them up and destroy them. Otherwise, your whole life itself as a cell is in danger. That much important. This correct folding and correct function, if you release not behaving correctly because of this misfunction, then you should face your own consequences. So you don't want to do that. Okay, that's one function. Another job that you go through as an editing procedure is once you are done a correct folding like this, okay? So you look good. You're looking good. And then what's next step? You put a little bit makeup, the base makeup on the surface of your protein. As a what? In the form of what? This sugar chain, what does that mean? Glycosylation. You are putting carbohydrate. The glycosylation means that attaching the carbohydrate is the process that we call glycosylation. And you're not done here, actually. Glycosylation is the first base makeup. You get more sophisticated makeup in your next destination. So this is the initial makeup in the form of this glycosylation. And once you're done, then now you're ready for being shipped to the next destination. How? In the form of this, which is called what? Secretory vesicle. The vesicle is the keyword. These two keywords. Secretory vesicle is the one. This isolated, independent, sac-like structure. Of course, you cut this thing in half to reveal the internal inside, but outside the actual or overall shape will be the ball-like shape. Okay? So this cargo, so to speak, is how do you make? You can imagine easily that what's the process of generating this secretory vesicle, right? How? The area, this membrane, composing this secretory vesicle was once what? A part of what? This material making this secretory vesicle was once a part of what? ER, yeah. So you push this cargo molecule toward the end and then squeeze, pinch them up. And that's how you generate a secretory vesicle. So this is simply just the same membrane, phospholipid membrane made the structure. And then you just push them into the next generation, the destination, which is Golgi. Here. Like here. So once you generate this secretory vesicle, and this vesicle will bump into the surface of Golgi. And the beauty of employing this so-called secretory vesicle as a transport vehicle is that all of them are made of the same kind of the phospholipid bilayer material. So they are all hydrophobic lipid molecule. Lipid will fuse. Lipid and lipid will be able to fuse easily each other. So you don't have any specific ritual for docking and transferring your internal cargo into the next generation. Simply you fuse them around like your invisible man fusing into this wall and then disappearing, and this transport vesicle can do so. And that's how easy is it. That's how you do this procedure. So you generate this vesicle and then make them fuse with the next generation destination. And once again, Golgi, when the next round of processings are done, Golgi also generate their own version of these vesicles, right? And then make them fuse out of the cell membrane. And some of them are facing Niagara Falls, like, oh, hello, bye-bye. And some of them somehow hung on to in the middle of. That's another a little sophisticated stuff. But since this is not a cell biology class, so I will skip that procedure. Simply, okay, some remain in the middle, some will fall out of the cell, and some are also shipped into another destination. And that's how you do it. That's the end of the story for this so-called exocytosis. Fine. Okay. So once again, so what's happening in the next destination, which is a Golgi apparatus? Let me compare this Golgi apparatus in your postal office. In your postal office, what they do is ship and sort. First, you receive those mailing stuff and then sort them out, and then according to their postal code, they send them out. And that's exactly what this Golgi apparatus does. Okay? So they can somehow distinguish. They are able to distinguish some different proteins that are shipped into the different areas. So like, oh, these guys are going to send outside of the cell, or these proteins are going to go to cell membrane, and these proteins are going to be shipped into the lysosome. They know somehow. And then they sort them out and send them. And that's their really the most important job. So protein sorting. Protein sorting is Golgi apparatus' function. And another, the secondary, more sophisticated makeup in the form of this glycosylation is also done in the Golgi. So once these proteins go through all the different layers, Golgis are stacked, just like stacked pancakes. Golgi consists of several different layers. So this protein has to go through all these different stages. They have to go all the way up. So once they are done, imagine how much more sophisticated makeup of glycosylation they must have received by them. So different protein will be able to have so many different variety of carbohydrate form. And that's how the outside of any recognizer are correctly able to identify different type of cell because of this. Okay, so that's so much for Golgi. Lysosomes. How many of you have some particular question about the previous exam question? Do you have any? Anybody have any specific questions? Or at this point you haven't, who even would start studying for the exam coming up next, way later, next Monday? I wouldn't. Of course, I agree. 100% agree. I wouldn't do that. I'm not the guy who actually do. I usually start on Saturday. Saturday. I still have a full 48 hours of Saturday, Sunday, and even exam is even on late Monday afternoon. Well, don't worry. And around the end of the Saturday, oh, still I have 24 hours of a Sunday. And at the end of a Sunday, then I usually get to think that, oh shit, I'm screwed. And that's how you do. In my case, of course. But anyway, I'm curious. Do you have any specific question? You don't. Okay. So if you come up with any particular question over the weekend while studying, you can text me by then. Okay, so then I can finish this lysosome business. Okay, so may I? Yes? Okay. So these lysosomes are stomach, acting like a stomach in the cell, which means digestive function. So all the garbages will be dumped or shipped into the lysosome, and lysosome will be gladly digest all of them. So everything, like you name it, protein, carbohydrate, lipids, and nucleic acid, and also those other bacteria. Even a lot of times, actually, some foreign bacteria are captured and then shipped inside of this lysosome, and they will get killed. And some viruses also try to invade into the cell, and then unfortunately the virus is captured, and they are shipped into lysosome, and they are all destroyed. That means this lysosome will contain so many such specialized enzymes, okay? Digestive enzymes. So there are so many different types of enzymes inside of a lysosome. Okay. Some specialize in protein digestion, some specialize in lipid digestion, some others specialize in carbohydrate digestion, and so you have all different kind of enzymes inside of this lysosome, and all of them have in common in terms of the chemical property is they are all they function at their best in acidic condition, just like the enzyme in your stomach. For example, in your stomach, there is a very famous protein digestive enzyme called, have you heard of this guy? Pepsin? That is the protein digesting enzyme in your stomach, and it's amazingly, their best optimal condition for this action is pH something like 2.0. pH 2.0 is just like highly acidic. Everybody will die in those pH 2. But this guy, pepsin, is their best environment. The other, more regular, I wouldn't say normal, but more regular protein digesting enzymes, their optimal action is usually neutral, like pH 7 or 8. So different enzyme, just like different cell that I earlier mentioned, that some bacteria can live in like 95 degrees Celsius and have no problem. So we stole the DNA synthesizing enzyme, DNA polymerase, from that bacteria, and then invented the PCR. So that guy who stole that enzyme became famous and also rich, right? Just like that. So different environment, different cell, and different protein will adapt like this. So here, the message that I'm going to convey is lysosome maintains also the same environment as our stomach, highly acidic condition. Inside of this lysosome is acidic. What's the advantage of being acidic in this type of action? Like why this both stomach and lysosome have this acidic condition in common, and coincidentally their functions are also identical in digestion. Why? Why is that so? What's the advantage of having this acidic condition? It helps like destroying. By acidic condition, all those materials will be easily destroyed, their chemical bond more. And so you have more advantage in complete destruction of any your target molecule. That's why. So anyway, but the more important question is how do you make this acidic condition is actually topic. Today's highlight. Why and how? How this lysosome and exactly the same. The reason why we'd like to pay attention on this lysosome is in your stomach, the way this stomach maintain this highly acidic condition is exactly, literally exactly identical to this mechanism. Yes?
근데 그 매우 강한 데서 사는 pH가 낮은 곳에서 사는 근데 펩신보다 더 낮은 pH에서 활동할 수 있겠네요.
Yes. Even some of the bacteria living inside our stomach can withstand such low pH. So it's just a matter of adaptation, like I said. So anyway, so how? This figure illustrates how you make the inside of a condition of this lysosome acidic. Yes? You're right. You don't? No, you don't. Using ATP and do what? Yes, you have to use the ATP, but that's a secondary. Proton, yes. So making acidic condition means you accumulate hydrogen. If you accumulate higher concentration of hydrogen, that means you make this area highly acidic. Acid base, the definition of acid and base. If you have a high concentration of hydrogen, then this environment becomes acidic. On the other hand, if you have much low concentration of hydrogen, then this condition, environment will become basic. So one of the quickest and easiest way of generating highly acidic environment is accumulating hydrogen ion like this. Hydrogen ion is called proton. So if you can somehow accumulate many hydrogen ion inside, okay, in comparison with the outside of the area, it always a relative things, then this area will become highly acidic. Here only it is showing only pH 5, but it can go further down if you keep doing this thing. Hydrogen. But the question is how? Why? What's the problem? The problem is, let's say you have a high concentration of hydrogen already. You have already accumulated enough of hydrogen ion, and then you still want more to import more of hydrogen being imported from outside into the inside, just like as this diagram shows, then you have a problem. Why? Because generally, naturally, all those material will move from the area of higher concentration to the lower concentration. That's the universal natural law. And you are now trying something against the law of the natural thing. So this action will it become spontaneous or non-spontaneous? This action, non-spontaneous. If you are trying the backward, exporting this internal hydrogen to the outside, it will become spontaneous because you have no problem. Okay, the only thing you got to do is just simply open up, open the sesame, then all those protons will flow out. But you are trying to squeeze more and more. You're being greedy. Okay, you stop it. You're greedy. So then, what do you have to do? You have to open your wallet. Energy. So that's why this energy is required. This type of protein transporter, protein transporter that requires the action of this ATP spending is called the pump. Any transporter that doing this, transporting things against the concentration gradient, all the material, all the molecule will move from according to the concentration gradient, which means from higher to the lower. That's what we call concentration gradient. Okay? naturally all those material, all the molecules will move from higher to the lower area, which means we usually say that according to the following the concentration gradient. Okay, understand? But sometimes you have to do backward. Then we call that as a against concentration gradient, which became non-spontaneous. Any job that you are trying that is non-spontaneous, you have to spend the energy, usually in the form of ATP. And those transporting protein that are engaged in such a non-spontaneous transport, we called pump. Why? Because their action resembles the pumping action using the energy is their generic name. Any non-spontaneous transporting function. So we do have, so this particular transporter, how would you name it? If you are able to name this particular transporter, how would you name?
K pump and A pump.
Pump is right, but would you rather call this as a K pump? Not the K pump? Potassium pump? Or proton pump? Because its job is delivering the proton. So yes, its name is a proton pump. And that same proton pump do also exist on the walls of your stomach, doing exactly the same thing, pumping proton from outside into inside of the stomach cavity. And that's how your stomach establish and maintain such a highly acidic condition, exactly as this lysosome do. Okay. One last topic before you go today is, so these lysosomes are involved in both. So any imported molecule through the endocytosis all end up in the lysosome, so that everybody are supposed to be digested in the cell, which is good. Although some viruses can escape this thing, they enter and they are supposed to go in to the lysosome, but they somehow learned to avoid and then bother us, but that's just exception. So anyway, so their function is digestion. And sometimes this lysosome do this thing called autophage. Phage means eating. Auto means self. So autophage suggests something like the activity of self-eating, cannibalism. Right? So autophage means, do I have, not here, okay, so I will finish the autophage over here. Autophage means when those, the old, aged organelles, especially mitochondria, if you keep operating such old, aged organelle, then it can have a higher risk of doing some harms, especially mitochondria. Mitochondria tend to generate a lot of these reactive oxygens. Which is deadly, just to exaggerate a little more. So reactive oxygen is not good for your health. So to prevent this, your cell actively pick up, scavenge some of those old, non-functioning, out of order organelles and then send them into the lysosome. That's what we call it autophage. Okay. So you get rid of those unnecessary, unneeded, and particularly potentially dangerous organelles. And also, another function of this autophage is when you are temporarily starving, autophage can help So let's say as a heterotrophic cell, unless you are provided from others, provided some food, then the cell, you as a cell, you cannot survive. And what if you didn't receive any food for days? Okay. Now, what are you going to do? Temporarily, you survive. You survive as a cell at the cellular level. You survive by eating up your internal structures like this. That's what you call the autophage. However, this autophage, have you heard lately the benefit of intermittent fasting? Have you heard of? It's called intermittent fasting. It's 2T or 1T, I don't know. But I always have a problem in spelling. But this intermittent fasting, which is this, everybody heard that this is good for your health. It will rejuvenate yourself. And actually, this is the benefit of this intermittent fasting is through this autophage. You skip your meal for a certain period of time, and then you kick on your cell, initiate this temporary process of this lysosome-based autophage. So by doing so, you preemptively get rid of some potentially old dangerous organelles like the mitochondria, and that serve as a potential benefit of. And then some people at least claim that, oh, because of that, I became younger, healthier. Okay, so probably that's the probably good point to stop here. So the exam will be up to this point, and so enjoy your studying, and also more importantly, enjoy your exam on Monday. Exam is nothing. Truly, I mean it. Your grade 10 years later, if you look back, you have this really 8th grade or failing grade on this shitty, miserable, unimportant class. What does that, I mean, it doesn't have any meaning. Only at this point, it may, to some of you, it may look much bigger than actually what it is. So, having said that, just don't be scared of anything, or don't be stressed out of any unimportant such business, and just simply break through and enjoy. Okay, so see you on Monday. Oh, by the way, next Monday exam is somehow I extended this reservation of this room. So if you want, if you are capable of, if you can stay more than 2 hours actually, if you want to finish up your exam. I hope, of course, none of them will need them, but if you happen to need them, you can